Welcome to the deep dive. Okay, you've shared some really interesting sources on sonication, also called ultrasonication.
Right.
So we're going to jump right in. Our plan is to unpack what it actually is, how it works, and maybe why it's such a go-to method for breaking open cells in some labs.
Exactly. It's a physical method.
Yeah.
Fundamentally, you're using very high frequency sound waves, ultrasound, within a liquid medium. And the goal is to disrupt cells. The real engine behind it, though, is this process called cavitation.
Cavitation. Okay, yeah, the sources hit on that hard.
Yeah.
Unpack that a bit. What's actually happening there?
Well, it's pretty wild when you think about it on a micro level. The sound waves traveling through the liquid cause microscopic bubbles to form, but they don't just float around. They oscillate, they grow, and then under the intense pressure changes from the sound waves, they collapse violently, like implode.
Wow.
And it's that collapse, that implosion, which generates these incredibly localized zones of high pressure, high temperature for a split second, and intense shear forces.
Like tiny shock waves.
Exactly. Tiny, powerful shock waves. And those forces are what physically rip apart the cell membranes or cell walls. We call that lysis.
Right, lysis. So the cell contents spill out.
Precisely. You release all the good stuff inside: proteins, enzymes, DNA, whatever you're after. So it's not the sound directly, but the physical force from those collapsing bubbles.
That makes a lot more sense.
Okay.
Okay, so the sources mentioned a couple of main ways to actually do this. There's probe sonication and bath sonication.
Yeah, those are the main lab scale techniques. The big difference is really how you apply the ultrasound energy.
How so?
With a probe sonicator, you have this metal tip, usually titanium, that vibrates at high frequency, and you stick it directly into your sample liquid.
Okay, direct immersion.
Right. It's very efficient because the energy goes straight into the sample. Great for smaller volumes. You get intense disruption. But there are downsides.
Like what?
Well, all that focused energy creates a lot of heat, which can be bad for sensitive molecules like proteins. You need good cooling. And there's a small risk. The probe tip itself can shed tiny metal particles into your sample. Contamination.
Ah, okay. So the bath sonicator.
That's the indirect approach. Your sample is usually in a tube or beaker, and you place that inside a tank of water. The tank itself is where the ultrasound transducers are.
So the sound goes through the water first.
Exactly. The energy transmits through the water bath to your sample tube. It's less direct, so generally less efficient than a probe. You might need longer sonication times.
But maybe less heat?
Generally, yes, less intense heating, and no risk of probe contamination. Plus, you can often process multiple tubes at once in a bath, which is handy.
Makes sense. And you mentioned continuous flow systems, too, for bigger jobs.
Yeah, for larger volumes, like pilot scale or potentially industrial, though scaling up has challenges. But basically, the sample flows past a sonication source.
Gotcha. So whether it's a probe or a bath, you need to control the process. What are the key dials you need to turn, the parameters?
Oh, absolutely. Control is critical. First off, there's the intensity, often related to the amplitude setting on the machine. Think of it like the volume knob. Higher amplitude means more intense cavitation, more disruption.
Yeah, more heat, presumably.
And more heat, exactly. Then there's simply the duration, how long you sonicate for. Longer time generally means more cells broken, but again, more heat, and also the risk of degrading the very molecules you're trying to release.
Right, you could shear your DNA or denature proteins.
Precisely, which leads to another parameter, pulsing, or the duty cycle. Instead of continuous sonication, you can pulse it like on for 1 second, off for 2 seconds.
Okay.
This gives the sample a moment to cool down between bursts of energy. The duty cycle is the percentage of time it's on. It helps manage that heat buildup.
So cooling is a big deal then.
Huge. You almost always need some form of cooling, like an ice bath or a circulating chiller, especially with probe sonicators, to keep the temperature down and protect your sample integrity.
Makes sense. Anything else?
Sample volume and concentration matter too. How well the sound waves penetrate and cause cavitation can depend on how much stuff is in your liquid.
It really sounds like a balancing act. Enough power to break the cells, but not so much you destroy the contents or overheat everything.
That's the perfect way to put it. It's a real balancing act.
So given that complexity, what are the main advantages? Why do people use sonication?
Well, when tuned right, it's just very effective. It works on a broad range of cell types, bacteria, yeast, algae, even tougher things like plant or animal tissues if they're minced first.
Versatile.
Very versatile. And it's relatively fast compared to some other methods like chemical lysis or mechanical grinding sometimes. Plus, sonicators are pretty standard pieces of equipment in many biology and biochemistry labs.
Okay, readily available. But what about the downsides, the limitations or headaches?
Heat generation is definitely number one. You have to manage it. Then scalability, taking it from a 10 milliliter tube to a 1,000-liter bioreactor, isn't straightforward. The physics don't always scale linearly.
Right.
Probe contamination, that small risk, is a concern for some applications. And practically speaking, they generate a fair bit of high-frequency noise. You usually want hearing protection or to put them in soundproof boxes.
Good point. So, considering the pros and cons, where does sonication really find its niche? Where does it shine?
It really shines at the lab bench and pilot scale for getting inside cells. If you need to efficiently lyse bacteria, yeast, cultured cells, or even break up small tissue samples to release intracellular proteins, DNA, RNA, or other biomolecules for analysis or purification, sonication is often a very good, reliable choice.
It's pretty incredible, though, isn't it, that the violent collapse of these microscopic invisible bubbles generates enough force to tear apart biological structures.
It really is. It's harnessing a pretty powerful physical phenomenon.
Yeah, it makes you think. You know, if these tiny cavitation events have such a significant biological impact, what other, maybe overlooked small-scale forces might be having unexpectedly large effects? Whether it's in other scientific fields or, heck, even in everyday stuff.
That's a great thought. It definitely highlights how manipulating things at the micro or nano scale requires this careful understanding.
It's that balance again, isn't it? Enough force to achieve your goal, but controlled enough not to cause unwanted damage. A real dance between power and precision when you're working with biology.
Exactly. A dance between force and control.